Right, so let's get started. Welcome back to our third day of the summer school. So this morning, we want to start with like the feature-based molecular networking job submission. So Robin and I gonna walk you through that. But before we do so, we want to reiterate on like this pipeline we tried to introduce to you this week. And as I pointed out a couple of times yesterday, and as Robin and Stefan showed you, like a very essential part of this pipeline is here, this data processing, besides acquisition, of course, what we did on Monday. So, and yeah, like this pipeline is basically structuring a little bit our week, right? So we did data acquisition and some, like general background info about like the mass spec data. Then yesterday we converted this data and then we processed it with MZMine. And yeah, today we want to move on to the next steps, which will be annotation. That will include like the feature-based molecular networking, which we submitted already yesterday evening. So hopefully most of you should have the done jobs by now. And then later on, Kai will walk you through Sirius, which you will see has like different like tools for in silico annotation. And then after that, Madeleine and Justin van der Hoof will showcase MS2 LDA. And then afterwards, we might move already a little bit into the visualization part, and I will show you how to merge this multiple layers of annotation within Cytoscape. But yeah, before we move on, I just want to like reiterate the feature finding yesterday. So who of you managed to get like the CSV and MGF output file and successfully submitted the molecular networking job afterwards? Okay, awesome. That's pretty much all of you who still had trouble and would need help to get to that point. Really nobody? Okay, one. All right, we should talk in the break and get you up to speed, because yeah, like basically this entire pipeline won't work well if you don't do the processing well, right? So this is really important to keep in mind. And yeah, so Robin is gonna showcase quickly now the concept. So after submitting the job yesterday evening, what actually happened under the hood in GNPS to get you to where you are right now.
Okay, so great to see you again. We did all this processing yesterday and it was kind of confusing, but as I already said, there's like faster ways to work with MZMine, especially if you know all the steps. You have the batch mode and you can directly run through everything in minutes. But in the end, we ended up with the aligned and gap-filled feature list, which is just like a feature matrix, basically, right? We have the samples, and then we have exported this so that we can submit it directly to GNPS. And there's multiple interfaces between the projects, and we're working together to make it easier. And we just now use the quick start or super quick start interface, which I think is like a very easy way to start with GNPS. But there's also then later ways how you can actually link up your data better and organize it better on Massive, because everything that runs on GNPS is then also stored in Massive. So what did we actually push to GNPS? We had the MS2 MGF, which was generated from the GNPS export in MZMine. It's a spectra file. So each feature that has an MS2 was exported and has now one spectrum, one MS2 fragmentation spectrum in the MGF. Then we have the feature table, which is just like a CSV with the area of each feature in each sample. And we submitted the metadata text file, yeah, so which is just the sample grouping, so that we can do some statistical analysis later on. The third file here is just optional. And what is happening under the hood is that we sent those features with retention time, mass to charge ratio, MSMS, and additional information. We send it to GNPS, and those features are then represented as nodes. So each of those nodes that you see in the network will actually correspond to a feature and an MSMS spectrum, right? And we create connections by looking at the spectra. So we compare them with a modification-aware similarity metric. For GNPS, that's the modified cosine similarity. So we just look at all the signals that are direct matches, but they could also be shifted because we're also interested in analogs, right? If we just have a molecule and we attach a methyl group on one of the side chains, whatever, it's going to be shifted by 14. And so the fragment signals could be direct matches, so we lose this 14 difference, or they could be neutral losses, so they could be shifted by 14. And then in the second step, GNPS is actually directly, and those numbers are actually outdated, so the library is growing steadily. GNPS is actually matching everything against the huge spectral library that maybe some of you already committed to and like added some spectra to. And if you have some spectra lying around, or reference compounds or compounds that you identified, you're always invited to share them with the community so that we can annotate more of our data. Because in those, like in this annotation step, you will usually see that maybe like 5%, maybe 10% of your data is annotated, depending, of course, of your study subject. And so if we now look at a bigger network, we can see all those edges, which are the modified cosine similarity, right? So the similarities of the fragmentation spectra, we hope are also in the similarity of the structures later on. So it's analogs of the compounds. And this is not your data set, it's a different data set, it's just an example. But you can see how different bile acids, and we have the free form and some conjugated form, are actually clustering together in some of the networks. And if we zoom in a bit more, we can actually see also, which is kind of obvious for some of the mass spectrometrists, that M plus sodium and M plus like protonated version, that they actually fragment differently, right? So that's what many people know. M plus H, so the protonated version, usually fragments way better and has way more signals than M plus sodium or M plus potassium, something like that. And why do we have those different adducts in the first place? Because during the ionization, of course, we can protonate, but we can also add like every cation basically to our molecule and ionize it this way, right? If we have some acid group, it's definitely going to be prone to capturing some of those cations that we have there. So those networks is going to be the basis of what we're going to work with downstream here. And most of the things going to work with this. And so this is very important to understand, like the concept of this network. And Danny's also going to show you how to make the most out of your network. There's not going to be one visualization that is like maybe satisfying you in all needs. You always have to like find the best way to convey the message that's like in the networks. And it sometimes gets very complex. So as this is so important, like, did anyone have some questions towards like molecular networking, modified cosine similarity, library matching?
So for now, what we look at today is only based on MS2. There is another step of like establishing connectivity between like the nodes, and that is on based on like the peak profiles. So yeah, that's a tool Robin and I developed called ion identity molecular networking that actually does like a feature correlation analysis between like, yeah, basically co-eluting compounds, and if they have like a very good matching shape, and if the mass delta fits to like some user defined values, you will also establish additional connectivity.
Yeah.
For the like, yeah, intro, we saw that this is like next level and might be like too complicated, but you will see if you watch some of like Robin's tutorials for MZMind 3, it's very easy to just set this on as next steps. So this is called ion identity molecular networking.
Yeah.
And basically brings together like this kind of like artifacts, what we see here, right? So this is kind of the perfect example. So you have sodium clusters for some molecules, you know, located elsewhere in the network, because due to like different like energy levels of these different adducts, you know, they fragment like perhaps like differently than some sodium or like protonated adducts, and hence they're like disconnected. And that's, I think was or is one of the bottlenecks of just MS2 based networking, and we hope that IIN kind of like improves that a little bit.
I would also like to stress that okay, we only see different adducts here, right? But you should also be cautious that you can also have in-source fragmentation. So I know someone who was always looking for fragile molecules, but then the in-source conditions like the drying gas and the declustering potential, everything was set too high. And so she was always losing her like glucose or whatever was on there. And even for the bile acids, we can see that free bile acids tend to have very intense, even more abundant M- water. So they have like water losses because of all the hydroxy groups in the aliphatic system. So, and then those are more abundant than the M+H that everyone is looking for, right? So you could get to some wrong conclusions just looking at like one ion. So it's always nice if you can find more. But
yeah, perhaps the same applies as like with the feature finding yesterday, right? Like, I mean, these are, I think, pretty fancy tools, but they're not showing you like absolute truths, right? So that's just one way of like looking at the data, and it's full of caveats and perhaps artifacts. So you as like the experts are kind of like needed to like be critical about what you find, right? And yeah, like the disconnection of like different adducts is one thing. Redundancy within the networks is something we see often, especially if your feature finding was not doing like a perfect job. So you may get like split features, right? So if you have spiky data, you don't do smoothing, and your deconvolution like tears apart features, then obviously you will get like multiple nodes in this network. Right now, are these like individual molecules? Perhaps not, right? So like I think you really like need to like understand what the tool actually does, right? And then be critical about it. And I think when used I don't know, responsibly, I think this can be really powerful. But yeah, if you just believe everything you see right on, then you may can go down like slippery slopes. All right, any more questions? Also to the feature finding yesterday. So we were happy to take some extra time, you know, just to make it clear what we did, and that we're all like on the same page there. Yeah.
When you establish a connection between the parts, do you just take into account the presence of some fragments, or do you also take into account intensity ratios and losses?
Yeah. So the cosine similarity takes into account intensity. So that's I think a essential part of the algorithm.
Yeah, there's some like pre-filtering that is done, and usually the intensities are like scaled. So you use like the square root of the intensity, so that like very abundant signals don't overweigh everything else. But yeah, then you have the cosine similarity. It's like a very simple metric, but it works pretty well for this kind of.
Maybe on that note, it's also important to stress out that that's also one metric and one way to look at. So there's more and more new matrices emerging, like spec2vec from Justin and others would be one. Or now the what is it,
MS2
like
deep score,
MS2 deep score, or spectral enthalpy,
yeah,
right, by the Fiend Lab. So, and I don't know what's like I guess none of them is like absolute, right? But they can be like complementary. So there again, like it's important to, you know, keep in mind that's one way of looking at the data.
We can say that most of the time, the modified cosine similarity is more towards like single modifications. And then of course, traversing the graph, we might have like a single modification and again a modification on a different side. And then, you know, you come to like multiple modifications over the graph. But other metrics might be better, for example, for like very complex multiple modifications. So,
do you want to go ahead?
By the way, the question for yesterday for that might be a short one.
Yeah.
I understood yesterday that NTMSS is taking the peak area, right? Not the peak height, which sometimes is called peak intensity.
Yeah.
Is it also possible to pick peak height in NTMSS?
Yeah. Me personally, I usually very often use the peak height because I think if you have usually like very narrow peaks and they don't vary between the compounds that you measure. I mean, of course, everyone knows some compounds, even though they come at the same retention time, are more sticky and then they like have weird shape. Then area is better, but usually if it's all like very narrow, I use the height. You can also export for statistics and for speech-based molecular networking, but just traditionally it was more like the area or GNPS.
So by default, the GNPS export module exports like peak area, but there is other export module where you can actually export a bunch of other like informations from your feature tables. Not only height as an alternative way of measure abundance, but also characteristics like the width of a peak, or like the width at like half height of the peak, like the I think shape you can report.
Like
so
there's
a
factor and stuff like that. So you can export everything if you want to.
So that's we didn't also not showcase it yesterday, but for like the advanced users, it's really convenient to have that information accessible. So that would be through like the export module, and then instead of export to GNPS, you would go to export to CSV, and then you can like actually check what information you wanna get out of it. Okay, there were some more questions. Yeah.
To this graph, is there any reason how they organize the nodes so if they're not connected to each other? Is there any chemical nature behind it? So it's just like white space and they put nodes somewhere.
Yeah. So if there's no connection, I mean, you have different we do it usually in Cytoscape. It's a very nice tool for like the graphical visualization of networks of graphs. But it's usually just a force-directed graph. So if there's no edge between them, there's no force. So they're just gonna disperse. And then there is MatGem, for example, where you can also export from Enzymine. You import in MatGem, and then they do the networking there. And they have like a TSNE visualization, and I think Tito is gonna show it. There everything has like is like in a 2D space, and but yeah.
Also there, maybe to add, there's a ton of possibility to organize your network in Cytoscape. And that is, I love that software too. So thanks a lot to the Cytoscape developers for making this. And there you can like have different like organization schemes of your network. So you can do like force-directed layouts, but you can also do other layouts. You can like organize this in circles, which may be like helpful to like better, like visualize like the, I don't know, like chemical complexity or whatever question you have. So we're gonna get a little bit into that in the last session today, and then in another one on Friday. So hopefully we'll give you a good intro to Cytoscape as well this week. Right.
So yeah, this might be a complete misunderstanding from my side, because I have a hard time wrapping my head around. This is based on the fragmentation data, no?
Yeah.
So you said it's just based on the MZ2. So in normal metabolism, a lot of the fragment, what happens in fragmentation also happens in normal metabolism. Can that interact? And you have, I mean, you think it's fragmentation, but it's actually just a metabolite that
I think
metabolic step has taken all the.
It's important to not mix up MS1 and MS2 there, right? In MS2, we isolate only one ion at a time and then fragment it. So if there is like already a pattern that would kind of like be natural from your metabolome, right? Then that would be all like individual peaks. Obviously, those should have similar fragmentation patterns. So we would hope that actually, like biotransformation products will be connected in the network. And I think that's exactly one of like the strengths, right? That we like pull like, like related metabolites together and cluster them in molecular families.
So it's kind of like the promise is already, we look at two different things. And we hope, of course, everyone here hopes it's like one known thing and one analog of a known thing, right? Which is like the new compound we want to discover. So we just already look at something that has a precursor MZ difference. So if we just look at the precursor MZ difference here, it's shifted by 14. Yeah, so we can then very easily conclude it might be just a CH2 of difference. We still have to, of course, figure out where the CH2 is located. It could also be something else, but so yeah, the hope is that it's going to be like two different compounds being connected in the network. And then, as I said, the graphs get quite complex, and then we can try to traverse the graph and make some sense out of what we already know and discover the unknown. Yeah. And this concept is very, I mean, even now, like I still understand it better after years of working with it. Like just last year was like, oh yeah, you can also explain the modification, like the 14 is also like a neutral loss. So because if you just flip the spectra at the precursor and then you look like, hey, wait, the signal's just shifted by 14. Oh, it's the same neutral loss. It's like we cut off the same branch of the molecule. That's why we keep the modification in the ion, and the other way around, we cut off the modification. So of course the parameters are the same, right? So there's still ways how we learn to maybe also explain it.
Okay, there was another question.
So when GNPS tries to figure out what's closely related, like what kind of does it depend on how diverse and complex the data set is? Because I feel like, well, you could say, well, they all have carbon, so they are closely related, right?
Yeah.
But so, and also, do I have any influence as a user on that?
Yeah, that's a very good question. Thank you. So yeah, like there was a couple of settings yesterday in this molecular networking job that we submitted that we did not really question, right? We just left them default. But you define, first of all, like the cutoff of like the similarity score by till what like value like nodes are connected. So by default, this was 0.7, but you can of course like increase or decrease that. And then you will get eventually more stringent, or you will get very loose and get like more false positive connections. And then there's another very important setting that isn't hidden under the advanced tab. So if you actually, I'll show you later how to get quickly back to the settings and change those for new jobs. There's a maximum connected component size where you actually define the maximum numbers of edges that one node can have. And that I think by default is 10, right? So, but now if you have a very complex data set where like one node maybe is like related to like many molecules, then simply once it reaches 10, it will like the lower scores will be cut off. And then also there's I think a maximum network size. So that's also like very critical for very complex data, or especially if you have like some chemical classes that have many variants, right? There, once like a subnetwork reaches a certain size, it will also cut off. That is to avoid a hairball effect. So if you do not have filtering there and like have a very low threshold, then simply everything will match somehow to like everything, and then you will just get like one hairball of a network, and that is hard to yeah, like make sense out of. So hence like there's like these values, but yeah, that depends on your data set what you actually want to use. I would suggest to start with the default value. So that's why we did this, and normally we get relatively good results with it, but yeah, you might want to look at this later on.
Yeah, the networks from the default settings are kind of like digestible, you know. So most of the very prominent connections, they will be there, and you're going to see, and you can go through the graph and make your connections like of those annotations. But yeah, like you could play around and just make it like, yeah, connect everything. Like you can make it more complex this way.
Always have to go back and restart the entire job for that?
Yes.
Kind of.
Yeah. Well, in Cytoscape, you could also lower, like raise the threshold by removing edges. But we typically just like rerun the jobs. And there's a, I'm going to show you in a second, there's a very convenient function in GNPS to do that quickly.
Yeah. And also talking to Ming, who developed GNPS, like he's like, yeah, just rerun it. Like it doesn't matter. Like it's like we have the resources to do that. So he's also rerunning everything. So yeah.
Okay. Was there another question? Okay. Awesome. Okay, one more.
It's a simple detailed question. For Capgemini, after I run, I try to save the project. I see there are the two different versions. Like one is simple and the
Yeah.
reference.
Yeah.
What's the normal way to go?
Good question. If you always process on the same computer, so the question was what kind of project style you should export from MZMine to being able to re-import. Me personally, I don't save projects. I just reprocess. I save the batch because it's usually so fast that I don't care. But yeah, for saving the project, you have the referencing style. So the raw data files need to stay in the location where they are, and then the project will reference to them. But that's for your local file system. If you want to share the project with someone, then we have the like complex create basically. Like we pull everything in, and then it's like self-contained. So you can send it to someone. It's humongous, and you can share it. But yeah.
Okay. Yeah. We hope with that it's a little bit becoming more clear, and I'm sure it will get more clear as we move on and actually start exploring this and doing more things with it. And then, yeah, anytime feel free to ask if something seems not clear to you. All right. So coming to like the big picture objective of today. So yeah, like how can we now leverage like the feature-based molecular networking to like increase like annotation, or like to provide annotation and interpret our data set. Okay, and yeah, so now we're gonna look at like a couple of different things. So first of all, we want to see, okay, what compounds did GNPS annotate for us? Then we want to look at like the quality of this matches by like manually inspecting them, and then we want to like perhaps start propagating already some of those annotations within like the molecular network. So we want to like look at actually connected nodes, and then I want to briefly showcase some advanced visualization features. So GNPS is much more than like molecular networking now, so you can actually do a ton of analysis in there, and it's really, I think, too much to like cover in even 1 week, but we want to get you started with it, and encourage you to explore later on more by yourself. One important thing, so there's this link down here, which we can also paste in our resources document. So there's extensive documentation for GNPS. So we put a lot of effort to make like a written documentation under this link, and it includes also like a lot of like videos and like recordings of past workshops we did. So there's, I think, quite a ton of resources online. But yeah, let's get now to our molecular networking job. So I would suggest all of you open this job. In case you do not have a job, I can just actually paste this here in the chat of the Zoom, and then also in our, I think there's a network also in our resources file. Right, so let's see. But if you have your own network, definitely look at that. Okay, so all of you have a network view now open that looks like that. Good. Okay, so then, like my first question to you would be, what did you find? So I checked already with a couple of people yesterday, and they found some molecules, which I found interesting and also encouraging, because I know already what should be in there. But now it would be interesting to hear what you guys found. And to like figure that out, you can simply click here on this view all library hits list, and then actually, you should get to like a table view where you see like like a number of like annotations. And let me actually just do that in the web browser as well. So again, yeah, we were lab 15 here, I think. So it might be, there might be some slight differences, but I think a couple of molecules should be very common in all of the data sets. So again, I just click here on view all library hits, and then it should load like a list with, I don't know, like perhaps like around 100 annotations or so. You'll see that quickly by like looking here on this hits thing. So here it says, it shows 1 to 30 out of 129. So we have 129 annotations here. Who can tell me what level of annotation this would be? Which one? Yeah, level 2. So this is matches based on MS similarity, right? So we don't call them identified yet. Okay, so what did you find? Who wants to? Huh, interesting.
Where you got yourself from again?
Well, the question is who sampled those samples? All right. I want to hear a couple. Come on, tell me. Yeah.
So there's this image here or whatever, is that a herbicide?
Okay. Who else found imazapyr? Okay, if you wanna, because there's 129 hits here now, and if I scroll down, it might be hard for me to see that. I can just like type in here imaz and see, oh, I found it too. Cool. So maybe if I found it and you found it, and who else found it? Okay, come on, not everybody, just a couple of people. Well, a lot of people. Okay, cool. So maybe that's if like all the labs, found it. Maybe it's it. Okay, what else? Carnic acid. Ah, yeah. Do you know what is this? Aha, yeah, it's a toxin. Okay, interesting. Who else found carnic acid? Oh, I got it too. Okay, you got it. Okay. Anthramoic. Oh, let's see. Oh, domoic acid and methyl domoic acid. Oh, interesting. So, DEET. Oh, yeah. Yeah. Okay, I have to be honest with you. If I see DEET, I'm always questioning, especially if it comes from fieldwork campaigns. People often use it. Yeah, I don't have it here. Which one? Mm-hmm. Like this, tree?
300.
Oh. No. But yeah, okay, let's see if I have cocaine too. Oh yeah, cool, nice, thanks. Thank you, Mass Bank. I got a very nice hit. So now, yeah, to like think already a little bit about confidence, whether I believe this or not. I mean, hearing it from you guys that you found it too, definitely add some confidence here. But also when I see that I have a cosine score from 0.98, 9 matched peaks, and a ppm error of 1, right? That typically means it's like a high quality match. Okay, so now how do I assess that, right? Like how do I get actually into the spectra? So there's a simple click here. You can just like, click here on view mirror match. And then hopefully, like this spectrum and like the matched spectrum will like pop up here and you see nicely, this is a really good match. I mean, I have 2 main fragments, but I also have a bunch of like the smaller ones, which might be pretty important for this high quality match.
In the mirror, the bottom one, the green one, is the library spectrum, so the reference spectrum, and then the top one is the one that we submitted for search.
Question?
So I have two hits at the for cocaine.
Yeah.
But I don't see I see some different numbers, but I don't see a different structure. Why is that?
Yeah, so that could be exactly that type of artifact I mentioned before. So if cocaine, the peak on MS1 level was like really big or had like a long tail and then some spikes, maybe the feature finding has split it in two. Or maybe there is a isomer, right? And maybe it is two peaks. It just has like the same MS spectrum. So like, I don't know about cocaine, but there's a couple of molecules that we actually want to see multiple hits to. And because we can't distinguish them by MS, for example, if it's like diastereomers or so, then obviously they should like match to like the same thing. Okay, so now, okay, that's one quick way to like look at the mirror match. There's also here like this USI links, and now I have to scroll farther down. Okay, let me minimize that other mirror match. So if I click here on USI links, then I see here also a mirror match USI and a query spectrum USI and so on. And that actually brings you to like the more fancy visualization tools. So let me try this. So now, yeah, like this opens up now this new viewer. And if you want to get like publication ready figures out of this, where you can actually define like the scales and export this as an SVG, so a scalable vector graphic for, I don't know, your high quality figure, then I would recommend you working with that. Yeah. So when I click here on this USI link, then there is like here this three links at the bottom popping up. And then I clicked on mirror match USI, and then like this new window appears, was basically like shows me the same thing, but now here I can actually like adjust a couple of things like the ratio of like the width and the height. And then for me, most importantly, I can export this as a SVG. Who of you knows what SVG is? Okay, yeah. So who of you uses Illustrator to make figures or Inkscape? Okay, so yeah, that's the vector graphic. And like that is, I would say, the go-to thing to actually make publication quality figures. Before here, I can only get this mirror match here as kind of like a screenshot pixel-based, which is fine too, you know, but like if you want to like put this in the middle of your manuscript, you may want to, you know, make the lines fatter, the labels bigger, whatsoever, and then you're quite limited. Like with SVG, you can like manipulate like all these things. I don't want to get into this now, but like a good portion of Friday will be actually dedicated on creating vector-based figures, not only out of molecular networks, but also out of like the other tools that we present. Mm-hmm.
Oh, the scores. Yes, good question. Good that we didn't miss it. So the thing is, this interface uses different filtering and also different implementation of the than GNPS molecular networking. Yeah, so it's kind of sad that we don't get the same scores, but here it's actually a process like when you demand it. So you can change the uses, or you can change the spectra that are used to any spectrum that's in the public domain. So that is on like metabolites and massive and on GNPS. You can change it to any of the spectrum, rematch it, and you can also specify if you want the cosine similarity, which is usually used for spectral library match, or the modified cosine similarity, which is for analogs with like a difference in the frequency.
Yeah, that's a really good point.
But it's not using all peaks.
No, I think here it is, but before in our GNPS job, it's not. And that's another thing that is under the hood there, hidden in like some of the advanced features, and that is like filtering of the spectra. So especially if you have instruments that produce like more noisy spectra, then like for like, yeah, calculating like cosine, like that small noise peaks kind of like are a little bit like annoying. So that's why they just have a shifted window in 50 MZ units and then remove like all like the low abundant peaks there. if I would turn this off, then I think it should like show you the same value. And actually that is something that you might want to try. So for me, working with like very complex data where I also often have chimeric spectra. So that means like spectra that result from different precursors in the same isolation window. You know, it kind of like is not beneficial there to like filter too much. So I get more hits when I turn this off. To quickly show you how to do that, if I would go back to like the original page here, I'm just gonna open this in a new window now. So yeah, when I'm back at this initial interface and I click here, clone to latest version, and I actually have to log in to do that now. So not logged in. Okay, let's try again. So if I go back to the initial view, and then there at the very top, there's this clone to latest version. That kind of like clones my job I submitted yesterday to like exactly the moment before I submitted it. So if I wanted to like test different settings, it's very easy to just clone, change the settings. I write in the description what I changed, and then I submit, right? So now here, you know, we did not open any of these advanced things yesterday. But if you go, for example, under advanced filtering options, you see that there's like some filtering precursor window and filtering peaks and 50 Dalton window. And then there's also a filter library. And that's exactly what was not done in this view here. So here, like the raw spectra have been mentioned. So now if I would turn this off, don't filter library, and yeah, okay, for now, let's just change this. And then I would just like resubmit this, not filtered, you know, submit. Then, well, we can check later if we actually get the same score then. But yeah, that's a quick way to like redo that. Also, now I can clone this again from here, clone to latest version. I think there was the question about the connected component size and so on. I also could quickly change this here under advanced network options. So this is the network top K. That's exactly the number of connections per node, right? And I can just bump this up to 20. And then the maximum connected component size, this is the maximum number of nodes in one connected network, right? If I would know, oh, I have like this bile acid family and there's like so many of them, way more than 100, then I can just increase that to like a number like 500. Also, now I can, as I mentioned, change the cosine threshold. So this would be this number here, 0.7. You know, I can like be more strict, you know, I only want to get high matching stuff. And then if I know that like the molecules I look at produce like a lot of fragments, then I can also increase the minimum number of matched peaks between two MS-MS spectra. On the other hand, if I know, oh, like I'm looking at these like really small compounds and they only have a couple of fragments, then maybe 6 might be too stringent, and I can lower this to another number. But yeah, again, so these are kind of like advanced things. That's why they're like hidden. But as you move on, like exploring this, you know, I would suggest to try some of these options out, right, and find the settings that are best for your data. And as you look down the line, you know, filtering I showed you, there's also different quantification options. So you can directly normalize your data, for example. You can like for some of the multivariate stats that GNPS does under the hood automatically for you, you can select different like distance matrices. By default, we have like here Curtis now, but you could do like Euclidean. So that would be the classic PCA, right? Or like a Jacquard or like cosine. Then for univariate, it also can like do some like univariate analysis directly for you. But then, yeah, you would need to specify here metadata information and so on. So really, I think for now, let's hide these and just get like started exploring the basics. And then as you move on, I would encourage you to explore. Okay, are there more questions to this point? Yeah, then I would just, you know, like, let's say here I change this now to 20 and 500. Then I would give this another description. So what was the top K, 20 and max component size 500. And then I just go down and say submit. And now it should run it. And once it's done, like yesterday, you get an email, or here under my job list, you know, I would see when it's done. Okay, another question? Yes, yes. So, I mean, downstream, I would obviously not remove that per se, because it might be different isomers or so that are actually real. But if you just want to get a quick overview and you say, oh, I have like, I don't know, an artifact with 30 same hits and I want to remove that, there is also this few unique library compounds here, just right next to few all library hits. And now if I would open this, then I would see that, oh, I actually have just 72 unique hits, which is significant less. So maybe there was quite some like redundancy within the network. And if you see like a strong difference there, that could be a good indicator that perhaps your feature finding settings were not like perfect, right? So like there again, peak splitting is something that we often observe. Yes. Yeah, that is something that, like, to be completely honest with you, I would not rely on very much, because that basically just tells you what user uploaded this. And like traditionally, there was at the beginning gold users from like the Dorstein lab that uploaded like a lot of like spectra. And now by default, any new user can only like upload bronze, right? But now, I don't know, there was a lot of like automated like library generation, and especially from like the QTOF platform that I don't think are necessarily as good as the spectra that I upload right now. But because of like the my user role, I can only upload bronze. So can all of you, right? And it's not necessarily gold. There is, when you go actually to the library hit, let's see, there is also a source described. Oh boy. Sorry.
Control Alt Shift H.
Yeah. Oh, it's passt schon, Chefs.
Zoom back.
So, danke. Yeah, when you go to like view library here, then it should tell you like some more things about this compound, right? And then hopefully it also tells you where this is coming from. Actually not the case here. So typically when you submit, let me see if I can update annotation. You can like specify whether this came from an extract or from like a purified compound and so on. And then also, if I have a compound that I'm not like 100% certain about, but I want to like add it to the library, I would like name this already into the title actually of the compound. I would say putative something. Oh, okay, that's, oh, maybe it's because I'm on Proteomics 2. Let's go to, yeah, when you open this, did you see that there is a source here? Because you went to this through GNPS. Yeah, just gonna open up the link we shared with you, so it's not on to Proteomics 2. So again, here, if I go to view all library hits, and then go to the view lib here. Huh, interesting. Yeah, it's not shown, but actually when you annotate spectra, then you can provide this information. So I just try to like update this now. And then, yeah, see here, there's compound source and it says it's isolated. I think it might be visible when you download it later, and then in the full output table, the source is visible. And then this can be changed to commercial. So this would be a commercial standard, right? Which is of course like more reliable than, you know, this is like something from a crude extract that has not been isolated, because that implies that, well, I mean, that likely was not like a level 1 ID at the first place. Yeah. So there is a lot of information. Another thing that is very handy, and I think we don't have ChemDraw installed here, but I get like Smiles code or Inchi code right from here, right? So that's why actually like a molecule is like drawn here. And if I copy and paste this into ChemDraw, it directly draws me the structure. So if you find an exciting molecule and you know, it's I don't know, it's huge, before you start drawing it in ChemDraw by yourself, you know, try and like get the Smiles code from here, or go to PubChem or so, where like the Smiles code is displayed. So that's often pretty useful. All right, another question?
Yeah, so let's go to change the settings to optimize it for our data.
Mm-hmm.
What is the criteria based on which you decide that this is optimized?
That's a good question. I don't have a straightforward answer for that, right? So like I would like base it on like some ground truths you have, you know. So for example, you have like 2 authentic standards in there, you know, then you know they're there, you want to make sure that we identify them. So in our case here, cocaine was actually an internal standard. So I spiked in a mixture of a couple of drugs actually, because we had them available as an authentic standard mix in the lab. So now if I hear that you found cocaine, I mean, it's funny too, but like it's good because we know that this was an authentic standard we added, right? So now I want to make sure that my settings find that, and if not, I may want to tweak them till I find it. And then if you have something that should be connected, right? So you know there is, let's say, benzoylgluine, which is a metabolite of it, right? You can like test, okay, is this actually connected in the network? And now if, let's say, I go to like the network view, or like to the cluster view, where I can like look at all nodes in this network, then I would go to cocaine, open up the network view, and I would check, is it connected to the other compound that I know? So that's, you know, one way, but it does not apply to like all chemical classes. So I think you're really like kind of like understanding like the principles under it, and knowing like your data set a little bit is important, but there's certainly no perfect answer.
So there's not based on any of the columns that you apply a threshold and you say, okay, this is let's say equivalent to items.
I mean, we already apply some threshold, right? So we already apply like the standard is cosine similarity 0.7.
Yeah.
Then 6 matched signals, which is already a lot for like very small molecules. We typically lower it to maybe 4 for very small ones, but then you should also be careful, right? Like you can also lower the cosine similarity, because you could get like true matches with 0.6 or something, but you also get way more like false positives. Yeah. So it's very hard, and you can have like false positive with 0.99.
So there is really, I think, best to like look at the matching spectra. I mean, obviously, the more hits you have, it's not like feasible anymore. So there is a couple of ways to estimate FDR. So with the Birka lab, we actually tested a tool that they developed called Pasatutu that like helps you like estimating FDR, but it's not super straightforward at the moment. So we're not there where proteomics, I think, is. But like, yeah, when we tested like a bunch of different data sets with the Pasatudu tool, we saw that typically FDR of, I think, like 5% was around like 0.7. And also just from like working with this data for a long time, that seems like a reasonable like cutoff. Obviously now, depending on your data set, that FDR like goes up and down, right? Like there was variance. So like for some, the 5% FDR level was like lower. For others, it was higher. So that also depends a little bit on your instrument and on like, again, like the complexity of and quality of the spectra you have.
And I mean, of course, like usually we do combine it with like some statistical analysis or something to actually also prioritize some of the nodes, right? And then we go into the network and we try to like propagate the knowledge that we have, because then at least we don't look at only one thing, like one feature with MS2. We can look at multiple and look at like how are they connected and what might be the relationship between the structural similarity. You mean redo? Oh, review, yeah.
Before you report this in the main text of your manuscript, I would strongly suggest you to do that.
Yeah.
In fact, I would actually put all the mirror plots of like molecules discussed in the main text into the SI, right? Be transparent. Or like with the USI, you can even share the URL. So with the GNPS dashboard too, like which we're gonna highlight later, because this is all in the cloud, you know, it's accessible. So I can now, you know, actually as a fun exercise, we can just do this here. You know, I can just like copy this URL and like paste it into our resource thing here. Wait. Oh, I can't write. I don't have write access right now. Anyway,
I don't have because I'm not.
So like, yeah, later as a fun exercise, we can just share it. And I mean, later on in the course, I actually want from you guys those links so I can like look at your project outcome, right? And the same way as I could throw that in here. Well, let's just make a copy and then I actually showcase you how I would do it. You know, like now I can paste this here as an example. And now let's share this with all of you. Link kopieren. I'll just throw it into the chat of the Zoom. How do I get the Zoom back, Robin?
Escape.
All right. So now if you open that link, then you should get to the resource file. And then if you like copy paste this long URL into your browser, you should be able to like not only like, you know, view it, but also like manipulate. And that I think is really like, I think high level of transparency. Okay. We took a lot of detours now. Let's get back and actually like visualize some of like the connectivity of the network. And again, like, so I just go back to like this initial view we had. And now in order to not only see like the library ID'd compounds, but like all the nodes of my network, I can click here on this view all spectra with IDs button. And now if you do that, then again, a long table should appear, but now, yeah, with like basically every node. So most of them will be unknowns, right? And you see already, there is the spec family like column here, and some of them, there is a few network button. So those are nodes that are actually connected in a network, and where you don't see that, that means that they're singletons. So those spectra did not have any similarity to others in your network. That is actually another indicator to me about like the quality of my network and of my mass spec data and of the feature finding. Because if most of the spectra that I observe are not similar to anything else, then that is an indication to me that either there was a lot of noise in these spectra and they're like kind of like low quality. So with QTOF data, we see that quite regular. or that something was not so great with like the molecular or with the feature finding settings. But yeah, now to navigate a little bit in here, we can like sort here by library ID, and then, oh, I see domoic acid is here. I guess a couple of you found that too. And then in order to explore the surrounding chemical space of domoic acid, I can simply click here on this view network button, and then a new tab should open with like the in-browser network viewer. And then, yeah, if I go down a little and make this a little smaller, then yeah, very nicely, I see a nice network, right? So there's a couple of like connected nodes. And then here, like the blue highlighted one, that would be domoic acid. Now, if I just click randomly somewhere here, like in the blue space, then like all of the nodes should be visible. Okay, so now, yeah, you may do not have domoic acid identified in your data set. Then just like take another one as an example and explore it. Who got domoic acid? Okay, half. So that also was an internal standard. So we spiked that. So maybe we could revisit a little bit the settings if that's not in there. But yeah, anyway, so like to move on from here now, I have basically like the chemistry that was observed by MSM and is similar to domoic acid captured in this view. Now there's a couple of things I can do. First of all, there's a number over each node. So this is like the row number of the feature table, or like here it's also called cluster number due to historic reasons. So that's simply like the index of like this feature. But now what interests me much more would be like the parent mass or like precursor mass. So under this node label tab, I can just like click on it, you know, and then I see, oh, there's actually the different masses. And then, yeah, cool. Like here's domoic acid. You know, if I would click here on library ID, then I would get the names of the ID'd compounds. So yeah, domoic acid. And then, oh, methyl domoic acid is like a neighboring node. So that has a methylation of one of the carboxylic acids. And now, yeah, that would be for me a good example that I say, oh yeah, okay, that makes sense, right? So like, at least here, like the networking did a good job. If I now also want to inspect this, I can just like left click on domoic acid and right click on methyl domoic acid. And then on this top and bottom view here for the spectra, actually the spectra will be displayed. And with like this little magic sticks here, I can even annotate like the masses, and I see, oh, okay, some of them shifted exactly by 14, which would correspond to the methyl group, right? So here, 266 plus 14 would be 280, right? So that makes sense. And then some of the lower MZ fragments here are the same. So yeah, I would say, okay, there is like some spectral similarity in here. Now, obviously, I can like also expand this to like the unknowns, right? So I can like look, oh, there is actually one here, 340 and 345, and another one, 326. So the delta mass, okay, I can calculate it, or I can just quickly click here on this edge label for delta mass at the bottom to like display the delta mass. Oh, they're all 14, right? So maybe there is more methylation going on of the carboxylic acids. Could be something. So in that way, you know, we can like propagate annotation through it. And now with like my chemistry knowledge of like this molecule, you know, I make him propose already a new structure. And like I did with albicidins, what I showed you, I think on Monday, right? Like I knew like the biosynthesis like pathway of it, and I knew like what potential like amino acids could be in there, and what like tailoring enzymes might modify that. So I can like just from that knowledge propose structures. And in our case, it was successful, and like we could show by total synthesis downstream that this was actually correct. And I think this is really powerful, because I don't need to purify something right away to get like an NMR structure, which, I mean, hands down at the end might be one of the options I have to ultimately prove it, but I get like suspect structures or like putative structures fairly quickly. So yeah, I think that is very useful. Okay, another question?
So again, in this in this case, I have 2 domoic acids and 3 methyl domoic acids.
Yeah.
Is that similar thing is okay?
That could be actually the case, because so there is a couple of regioisomers of domoic acid. So I would not be surprised if there should be multiple chromatographic peaks, but it would kind of like ring a little bit of a bell. And I would definitely look now at the XICs of those and check, oh, were they actually defined features, right? And I can like nicely see them where they're like separated peaks in the chromatogram, or was that some sort of like artifact? So that's really when you need to go back to like the chromatograms too. I'm not going to do this right now, but there's an easy way to do that from GNPS, or if you still have your MZMIND project open, obviously you can go back there, or use Qual Browser or whatever other visualization tool you have. Yeah, another question?
Yeah, I have question. One is one substructure in GNPS, and the second one is, can we locate this in which sample this component?
Yeah. Yes. So for feature-based molecular networking, right here out of the network view, there is not. If you do classic molecular networking, you can, because you can subdivide this in this default groups here, and then you can label from which group it came from. We mainly use feature-based molecular networking as an annotation way, and then do like the rest downstream. So I would now import all like this information into my feature table, throw it into Cytoscape, and then make this pie charts in Cytoscape. I'm gonna show you that like in the evening. So yeah, from here, I would not recommend doing that, but rather do it downstream. And then the same for like the Planck subtraction. I think with like the classic molecular networking, you can do that. For feature-based, I don't know, and I would also not recommend. Yes. No. Yes. And then now we, yeah. Exactly. And then you have in your feature table the different columns for the for like your samples. Then you will group them downstream, calculate like a mean or average, and then you would build like the ratio. And some of that we will do in the univariate analysis tomorrow with Philip. So, but yeah, that happens downstream. Right now, let's see this as a, well, first exploration. So I wanted to show you what you can do here, but now the next step is to actually pull this down and, you know, what was it? Alt?
Alle modify on top. Control Alt Shift.
Thank you. All right. So yeah, let's go back to the to our network view here, to our initial view here really quick. And now I'm going to show you briefly how to actually get it down from GNPS and download the data. So there is here this download Cytoscape button, and I just like click on this, and then a zipped folder should be downloaded onto my computer. And now all the information is there in multiple tables. And okay, let's copy this into our summer school folder here. So here, yeah, it's a zipped file, so I need to extract this. And then, yeah, I get here like multiple folders with all the information GNPS made for me. And now one very important one would be like the GraphML file, because this is what I drag and drop into Cytoscape. So here is like this .graphml file. And in the Cytoscape session later on, we will go on from there and throw this into Cytoscape. But then there's also this DB results, which stands for database results. So here, this is a TSV file. So I can actually drag and drop this into Excel, or if you have open office into your spreadsheet tool there. So let's see if that works. So yeah, I just drag and drop it in here, and now I get like the full information. And I think to like unlock like all the infos, that is how I would like work with it. And now let's see, I think there's now much more information visible than in the initial view. So now we also have like some classes here. There's like, yeah, like the Inchi key, and there should be the smiles somewhere too. And then wondering if the source, yeah, nice. So now also here we have now here this compound source, right? So that was, I could not actually see this in the network online viewer, but now here it is available. Then like the, yeah, precursor mass, then like the calculated mass, the charge. There should be my mass error. So this is a very important thing. And then obviously the cosine, which here is called now MQ. So again, apologies for like the confusing naming, but yeah, like due to historic reasons, it was hard to like to change that then, and I don't know, make it uniform to cosine or so. So here it's MQ. All right, and then now I have also somewhere the actual feature table. Let me show you that. So here, if I would go back, there should be like the quantification table and the reformatted quantification table. And now, okay, this is a CSV file. Again, I can just open this in Excel, and I can like now, you know, simply look at it, and that is all the quant information that comes in from MZMine. So that should be very similar to what you exported yesterday from MZMine, right? And then here you have like all the samples, and then in like each of the fields, the peak areas of a particular feature that is listed here, right? So now I would need to find like the domoic acid feature, and then I could do any univariate analysis in here. Right. Yeah. So this is the overview of all the folders you downloaded. Oh. So there is the download cytoscape data button. So on this very first view we went, you can download it from there. And then, yeah, get the data. So yeah, you will see there's a ton of different things, right? Again, much more than we can actually cover here. So like, feel free to explore. For example, you can download also Ampere plots from like actually like the PCOA. So if I click here on, or it might be even in the, let me check. Yeah, it might be even here already in my folder structure too. So there's this GIME2 output files. And if I open that, then you will see like this QZA and QZV files. And those are actually visualization files for like the PCOA plots that GNPS made for you directly while processing all that stuff. So instead of like the analysis that Madeleine and Francesco are going to do with you tomorrow, like I don't think that this is kind of like a final thing, you know, because there was no blank subtraction. It's just a brief overview, but it's very convenient, you know, to just like have this available and then drag and drop it into like the GIME2 viewer and check out, hey, how was like my clustering in PCOA space, right? So there's, as I said, a ton of information. I quickly show you the GIME2 because I think that's really cool. So you just go to GIME2, I think it's .view. Let's see. No, it's view.gime2.org. And yeah, there's this drag and drop field, and any QZV file you can just like drag and drop in there. Let's say I wanna check out here the Emperor QZV. So that would be the PCOA plot. Then, you know, I can throw it in and I have a PCOA, right? And now here I see, oh, there was actually a lot of variance explained on like my PC1. That's probably the Planck. I wanna check this out. Sample type. Okay, here, sample type. Yes, red is the process Planck. And then I have my samples here. Obviously, it does not make much sense to like make this like PCOA with like the Planck in there, because most of like the variance again is like here on axis 1 between like my samples and the Planck. But you can already get like an idea, oh, there is some clustering also between the samples, right? So now if I would use time points, oh, then I would see nicely that here from 0 to 5 to 15 to 45, I see like a nice transition in PCOA. So now, just as a general check, even though again, this is kind of like not final because the Planck is still in there, but it gives me confidence that, well, what we observe here kind of like fits a little bit to like my biological expectation, because I saw it, you know, in our like dilution series, we would have like that Synechococcus popping up, you know, and then like growing and taking over a little bit like the community, and then also like the chemical space, right? So we would expect like a transition from like, you know, like the 0 time point to 45. And that's pretty much what I see here, right? I would now draw a line between like those plots, then that's the trend I would see. And obviously I see very strong clustering within the 3 replicates, which is not surprising because those were technical replicates, not biological replicates. So that's also what I would expect. The Chime 2 Emperor QZV. And oh yeah, there's a bunch of other ones. So there's also the biplot file, so you can try that too. That displays you already like some of the driving features. And I think there's also directly a Permanova. Yeah, there's a Permanova done. Let's try that one too. So, you know, it tells you, okay, is that clustering significant? And then we would see, oh, here it's not. But that was, I don't know. Yeah, so, I mean, it depends. This is, again, it's the first overview, and I don't know exactly what I set up in the metadata for there, but yeah, so you can get some infos fast. Again, tomorrow, Madeleine and Francesco are gonna walk you through exactly the same analysis in R, and they're gonna explain this way more in detail. But just so you know, you can do that also quickly in GNPS. Okay, question? Exactly. Exactly. Yeah. So that is what we take out of it, like as the main take home, right? I want to get network connectivity between my features of my feature table and annotation. And that's kind of like at the end all we take, and we move on from there, and then we add Sirius and MS2LDA on top, right? And then what we need is just like the feature number as our kind of anchor that, you know, connects like that. And that is given from like your output table from MZMine yesterday. So now there's this row number, and that is the feature number. And that is kind of like the, you know, identifier throughout all our analysis now. And that comes with a mass and a retention time value, but now we will add other values on top of that, such as like, you know, library ID. Okay, I guess that was our main take homes from like the feature-based molecular networking so far. Again, later on, we will take this. So what you just downloaded, we will take and then visualize on Cytoscape at the end of the day.